Nematology study of free-living and plant parasitic nematodes is known as nematology. Nematode, it derived from two Greek words, nema threadoids equals like means thread-like organisms, also known as eelworm or roundworms, because round in cross-section. Definition: nematodes are triploblastic, bilaterally symmetrical, unsegmented, pseudocellumatic invertebrates, with a true radial esophagus, and lacking specialized organs for respiration and circulation. Nematodes are pseudocelomate. Only ectoderm is linked with the mesoderm, while it is not lined to endoderm, or alimentary canal of nematode. Morphology. The nematode body consists of a tubular structure, having two tubes, i.e. the outer tube is known as body wall, and the inner tubes is known as alimentary canal. The space between the two tubes is called as body cavity, which is pseudocoelom, because it is devoid of mesodermal linings. The body wall, outer tube, of nematode consists of three layers, i.e. cuticle, hypodermis or epidermis, and the muscle layer. A. Cuticle: it is non-cellular, non-living layer of outer body wall. It is made up from structural protein, i.e. chitin, is not found in nematode cuticle, while in insect, cuticle is made of 25 to 60% chitin and 25 to 40% protein, on dry weight basis. B. Epidermis: the epidermis of nematode is unicellular layer, and its major function is to secretion of cuticle. C. Muscle layer, internal layer, which muscles are attached to the alimentary canal. Inner tube of nematode body is divided into three parts, i.e. stomadeum, mesenteron, proctodeum. 1. Stomadeum. It is the anterior parts of alimentary canal. In plant parasitic nematodes, PPN, the stoma walls form hypodermic needle-like structure known as stylet or stomatostylates. 333. 2014. In Thailandshid, PPN, stomatostylate is comprises of three parts, i.e. conus, shaft and knobs. Unit 2 Helminthology study of nematodes, which are parasitic exclusively on man and domesticated animals, is known as helminthology. Animal parasitic nematodes, APN SN 123 nematode, APN, placenta gigantissima, Dracunculus medanusis, Wuchereria bancrofti ascaris lumbricoides. Enterobius vermicularis onchossera volvulus disease. It is largest APN, about 8 m long, found in whale fish guinea worm, cause narubala disease in human, cause elephantiasis, or filariasis disease in man. JIF Ento 11, the vector for filariasis disease is Aedes aegypti, mosquitoes. RPSC AAO09, known as roundworms, intestinal parasite of human. It causes severe itching in anal area, particularly while children are sleeping. It causes onchocerciasis or river blindness, commonly in Africa, transmitted by black fly, Simulium sp. Unit 3 History of Nematology 1743, J.T. Needham, discovered first plant parasitic nematode, PPN, Anguina triticea, and it was named as Vibrio triticea 1855, Barclay, reported root knot nematode, Meloidogyne spp, from roots of greenhouse cucumber in England. 1857, Kuhn reported stem and bulb nematode Ditilencus dipsii. 1859, Schacht, reported sugar beet cyst nematode, Heterodora shachti. 1865, H. Sebastian, he known as father of nematology, and wrote monograph of the Anguillulidae. 1871, Kuhn first used CS for control of sugar beet cyst nematode. 1884, J.G. Demon, gave biometry and taxonomic monograph, Morphometry. 1907, N. A. Cobb, known as father of American modern nematology. 334 456 2014. 1933, Tom Godey, he published two books, 1. Plant parasitic nematodes and disease they cause in 1933. 2. Soil and freshwater nematodes in 1951. 1937, Chitwood and Chitwood wrote a book, An Introduction to Nematology. 1943, Carter discovered nematocidal properties of DD, JRF09. 1945, Christie, discovered nematocidal properties of EDB. 1956, Nematologica, first journal of European Society of Nematologists. 1958, Hewitt, Rasky and Gaheen, first reported the transmission of viruses by nematode, Ziphinema index, transmitting grapevine, fan leaf virus disease in USA. 1961, G. Thorne wrote book Principle of Nematology, known as Bible of Nematology. History of Nematology in India, 1901, Barber reported root knot nematode, Meloidogyne spp, infesting tea in Kerala's first PPN in India. 1913, Butler, reported Euphra disease of rice caused by Ditilencus angustus from Bengal. 1919, Milne, reported Anguina triticea, seed gall, of wheat in Punjab. 1936, Daster, reported white-tip disease of rice by Aphelenchoides hessei. 1958, Vasudeva reported Moliodiasiase of wheat, H. Anini, from Neem Ka Thana, Sikar, Rajasthan, later by Sisadari, 1959. 1961, Siddiqui, reported citrus nematode from UP, Aligarh. 1961, F.G.W. Jones reported potato cyst, Globodera rostokiensis, nematode from Nilgiri, TN. 1966. Division of Nematology established at IARI, New Delhi. 1969, Nematological Society of India was founded at IARI. 1971, first time Indian Journal of Nematology published. 1977, All India Coordinate Research Project, AICRP, on pests of crops and their control started. Nematode Unit 4 Classification of nematodes. The word taxonomy coined by de Candolle in 1875. Phylum equals Nematoda class. Two important classes, Sesonentia and Adenophoria. A. Sesonentia, known as Phasmida, heavy checkmark phasmids are present. 335 2014, caudal gland are absent. A competitive book of agricultural class Sesonentia has two orders, namely Tylenchida, Aphelenchida. 1. Order, Tylenchida. Tylenchida is highly evolved and sedentary parasite in nature. It includes suborder Tylenchina. Tylenchina is having superfamilies of Tylenchoidea, Haplomoidea, and Heterodoidea. A. Tylenchoidea, families, Tylenchidae, for example. Tylencus, Belanolamidae, for example. Tylenchorhynchus, Belonimus, Anguinidae, for example. Anguina, Ditilencus b. Haplolemoidea, families, Pratolenchidae, for example. Pratolencus, Rhadophilus, Nicobus, Haploimidae, for example. Haplolaemus, Rotolenculus, c. Heterodoidea, families, Heteroderidae, for example. Heterodera, Globodera, Meloidogenidae, for example. Meloidogyne, b. Class, Adenophoria, a phasmida. Heavy checkmark phasmids absent caudal glands are present 1. Order, Dorolaemida. Superfamily, Longidoroidea family, Longidoridae, e.g. Longidorus, Paralongidorus, Xiphonematidae, e.g. Xiphonemia. Superfamily, Trichodoroidea family, Trichodoridae, e.g. Trichodorus, Paratrichodorus, difference between Tylenchida and Dorolaemida. Tylenchida-Dorolaemida presence of 3 parts of esophagus, 2-part esophagus, is present buccal cavity is known as stomatastylate buccal, is known cavity, as odontostylate cuticle possess, distinct annulations, or transverse striations cuticle annulations, is not clearly observed 336. 2015. Unit 5 Parasitism of nematodes 1. Ectoparasite, living and feeding on a host from outside migratory parasite for example. Xiphonemia, JRV 2010, Longidorus sedentary e.g. Hemicycleophoro. 2. Semi-endoparasite, migratory c.g. Haplolaemus, Tylenchicus sedentary e.g. Rotolenculus, Tylenculus. 3. Endoparasite, enters the host tissue and feeds from within. Migratory c.g. Pratolencus, Rhodophilus. Sedentary e.g. Meloidogyne, Heterodera 4. Both ecto and endoparasite, Ditilencus aphelenchoides. Anguina Unit 6 Important plant parasitic nematodes. 1. Seed gall, ear-cockle nematode of wheat, Anguina triticea 2. Tundu sehu or yellow ear rot of wheat, Anguina triticea plus Clavibacter triticea. Yellow slime or gum on leaves is major symptoms of Tundu. 3. Cereal cyst nematode, Heterodera aveni. Root knot nematode, Meloidogyne spp. Citrus nematode, Tylenculus semipenetrans. Formation of 6 to 10 cortical cells, known as nerve cells is specialized symptoms, cause slow decline or dieback of citrus. 6. Reniform, pulse nematode, Rotolenculus reniformis. 7. Stem and bulb nematode, Ditilencus departure sad smiley onion bloat in onion is major symptoms of D. Dipsii. 8. Stem nematode of rice, Ditilencus angustus. Cause euphra disease of rice. 9. Potato root nematode, Ditilencus destructor. 10. Mushroom nematode, Ditilencus mycelophagus. It reported as first mushroom nematode. Second mushroom nematode is Aphelenchoides composticola 11. Bud and leaf nematode, Folia aphelenchoides sp. Rice leaf or white tip nematode, Aphelenchoides basaei. 12. Coconut red ring nematode, Rhydonaphelenchus cocarphilus. 337 2015, dr. Vector for this nematode is palm weevil, Rhynchophorus ferruginensis. 13. Potato cyst nematode, Globodera rostochiensis. A characteristic symptom syncytium developed. 14. Root lesion nematode, Pratolencus sp. JIF 2011, also referred as meadow nematode. 15. Rice root nematode, Hirschmaniella sp. Cause Mentec disease of rice. JIF 2011. 16. Burrowing nematode, Rhodophilus sp. Infective stage, all 17. Blackhead disease, rot root or rhizome root of banana, Rhodophilus similes, also cause yellow pepper disease in black pepper. 18. Spreading decline of citrus, Rhodophilus atrophilus. 19. False root knot nematode, Nacabus sp. Because symptoms resemble to as root knot nematode by enlargement of cells. 20. Stunt or stylet nematode, Tylenchicus sp. 21. Ring nematode, Criconema sp. 22. Pin nematode, Paratolencus sp. JIF 2011. 23. Needle nematode, Longidorus sp. JIF 2011. 24. Dagger nematode, Xiphonemia index. JIF 2011. 25. Stubby root, Trichodorus sp. 27. Lance nematode, Haplolemus sp. JIF 2011. Unit 7 Common crop nematodes 1. Seed gall, ear-cockle nematode, Anguina triticea first reported by J.T. Needham, 1743. He called Vibrios, name Anguina given by Philip Jev in 1936. In India first reported by Milne from Punjab in 1919. This nematode caused epidemic in Bihar, MP where the farmers burnt the standing crop of wheat. This nematode association with the bacterium Clavibacter, Corynebacterium, triticeus caused yellow ear rot or Tundu disease of wheat. The seed galls have 3,000 to 12,000 second stage juveniles, J, in a quiescent stage it first feeds, as ectoparasitically, and after 3 molts it becomes endoparasitic. 338 ter 2016. Some species of Anguina produces galls in leaf surface for e.g.a. tumefaciens on Synodon transvalens. JIF 11. Symptoms, seedling leaves are twisted and crinkled. Seedling may stunted and die. Plant show profuse telling, and infested plant produce ears almost 30 to 40 days before healthy plants. Finally seeds are replaced by galls. The most important character of this nematode is the conversion of grains into tocarcles or galls. Management. Salt flotation, use of 20% salt, brine, solution for washing of seeds is very effective control method. Hot water treatment, 54 degrees Celsius for 10 minutes. Crop rotation, also useful method to escape infestation of nematode. No resistant variety of wheat are found against this nematodes too. Cereal cyst nematode, Heterodera aveni cause mollier disease in wheat and barley. Heteros different and des skin means different types of skin, and lemon-shaped female. Females have zigzag pattern, or lace-like pattern on body known as cyst wall pattern. Cyst nematodes, includes genus, Heterodera and Globodera have unique character of retention eggs and larvae within mature female body which turn into brown or black hard body after its death, that why known as cysts. Syncytium is formed at feeding site in cyst nematodes. Cyst nematodes are generally host-specific. Pigeon pea cyst nematode, H. Kajani, also infests other pulses. Sugar beet cyst nematode is Heterodera scatii. Soybean cyst nematode, H. Glycine, it causes yellow dwarf disease in soybean. Heterodora orosicula, paddy cyst nematode, serious pests of rice in Kerala, Orissa, Haryana in upland rice growing areas. Damage. J is infective stage ETL equals 3 eggs per g of soil. Management of cereal cyst nematode, 339 2016. Crop rotation with mustard. Chickpea is most effective control because it is very host specific. Early sowing escape to damage deep plowing is destroyed. Quiescent eggs of nematodes grow resistant varieties CG for wheat. Raj Mr. 1 Rajkaran for barley. Road 2052, Road 2035, and Road 383. JIF 11, 3. Potato cyst nematode, Globodera rostokiensis. Golden nematode of potato first reported by FGW Jones in 1961 from Nilgiri Hills, TN. It feed pericycle cortex with results formation of nerve cells i.e. syncytium develop, which provides nutrients to female throughout. Development. Management. Legislative methods. Domestic quarantine was introduced against Globodera, not carry-infested potato tubers without phytosanitary certificate issued from Nilgiri to other part of country. JIF 06, resistant variety, Kufraswana, is most resistant to Globodera sp. Fumigants, methyl bromide exposure for 5 to 6 hours. 4. Root knot nematode, Meloidogyne spp. Means melon or apple-shaped female. First reported by Barclay 1855 from cucumber. In India it reported by Barber in 1901 on T, Kerala. In Meloidogyne the perineal pattern present on vulvoanus area, which is helpful in species identification. JRF 07, eggs lay in gelatinous matrix. 200 to 500 eggs. Sex differentiates occurs in late second juvenile stage, J, where the genital primordial is become V-shaped in female and I-shaped in male. Second juvenile, J2, is only infective stage in Meloidogyne. JIF 11. Spiketail in Meloidogyne, it is succeeding molting of second, third and fourth stage juveniles of Meloidogyne spp. Sexual reversal, in Meloidogyne sometime under adverse conditions male changed in toph male. Most of species of Meloidogyne completed life cycle within 25 days at 25 to 30 degrees Celsius and 7 to 8 overlapping generation completed in a year. Classification. M. incognita. Present throughout India on vegetables. 340 2016, Ichatrol M. exigua found on coffee in Karnataka. M. hapla M. Arenaria, restricted to temperate parts, on hilly area. It is a serious pest of ground nut in Gujarat. JIF 11 M. brevicorda, found on tea in Assam. M. graminicula, found on rice in Kastan part of country. JIF 2010, 261 symptoms. The reaction to the entrance of the larvae is the development of hypertrophy, enlargement of cell size due to nucleus division of cortical cells and forms 6 to 10 giant cells from which it derives food. Nematodes secrete protease enzyme results break down protein into amino acid, tryptophan, which is a precursor of IAA, is important and responsible for creating hormonal imbalance at site of reaction. Due to excess IAA formation at feeding site cause gall not on roots. Difference between root knot and rhizobium nodules. Root knot in case of Meloidogyne infestation, root knots are hard in consistency and deep-seated, they don't easily rubbed off management. Rhizobium nodules are spongy consistency, found alongside of roots, as side appendages soft and can be easily detached from roots. Deep plowing in May-June is very effective to kill quiescent J stage. Interculture of marigold, Tagetes sp, as a trap crop. Grow resistant varieties. 4. Tomato Hisar Lalit, SL 120 for brinjal, black beauty, Nilkanta. 4. Chili, Pusa Jwala. Biological control of Meloidogyne, fungus, Pisolomyces lilienus bacteria, Pasteuria penetrans both have been reported as promising by control agent. BT Kostaki, also effective against Meloidogyne sp. 5. Citrus nematode, Tylencula semipenetrans reported by Cobb, 1912, USA. Heavy checkmark in India by Siddiqui, 1961 in Aligarh, UP. 341 2017. Heavy checkmark juvenile second, J2, is infective stage. At feeding site forms 6 to 10 nurse cells. JRV 11, it is cortical feeder, as sedentary semi-endoparastic. Symptoms, slow decline as citrus decline and dieback. JIF 2011, 6. Reniform nematode, Rotalenculus reniformis heavy checkmark mature female, is kidney-shaped. Reported by Linford and Oliveria from USA in 1940. Heavy checkmark in India by Seshadari and Shivkumar, 1963 on cotton. Heavy checkmark pre-adult, immature female, is infective stage. 2 races, race A multiply on cowpea, castor and cotton. Race B reproduces only on cowpea, JRF 11, 7. Stunt or stylet nematode, Tylencohincus spps. Migratory semi-endoparasite Tylencohincus brassicae, feeds on cruciferae. T. brevilaneatus, cause Karlhasta disease in ground nut in APT, vulgaris, feeds on cereals. T. claytoni, known as tobacco stunt nematode. 8. Stem and bulb nematode, Didylencus sp. Nemawal, are frothy substances secreted through saliva of pre-adult, J. nematode responsible for quiescent in Didylencus. A. Rice stem nematode, Didylencus angustus in India only D. angustus is reported. It is reported by Butler, 1913 it causes euphra disease infestation mainly in deep water paddy. Symptoms, twisting of leaf and leaf sheath. The symptoms of euphra are well noticed in 2-month-old crops. B. Stem and bulb nematode, Didylencus dipsire heavy checkmark by infestation of this nematode onion suffers heavy losses, as it cause onion bloat disease. C. Potato rot nematode, Didylencus destructate found in temperate region of Europe and N. America cause rots on potato roots. D. Mushroom nematode, Didylencus myceliophagus this nematode was first reported that to cause damage on mushroom. It is also found in India. 342 equals E 2017 Ultra. Also known as foliar nematode 9. Leaf and bud nematode. Aphelenchoides sp. 1. White tip or rice leaf nematode, Aphelenchoides basae feed actoparasitically this nematode associated with serious epidemic of rice in MP. Heavy checkmark humidity favors infestation. This is seed-borne nematode chrysanthemum, a foliar nematode, Aphelenchoides ritsemahosi heavy checkmark known as spring crimp or strawberry nematode 3. Aphelenchoides composticola important nematode of mushroom and known as Mycophagus nematode 10. Red ring nematode Radonaphelenchus cocarphilus cause serious disease of coconut palm, is called red ring heavy checkmark. This nematode is transmitted mechanically through palm weevil, Rhinocarphorus palmarum. 11. Root lesion nematode, Pratolenchus sp. Also referred as meadow nematode. 12. Rice root nematode, Hershmaniella sp. This nematode responsible for causing Mentek disease of rice in 1949. Adult infective stage, all juveniles, J, and adult also. JIF 2011, heavy checkmark. It can be managed by soil treatment of carbofuran at 4 kg, a.IR before sowing. 13. Burrowing nematode, Rhodophilus sp. A. Root and rhizome rot of banana, Rhodophilus similis. It is commonly found in India. Heavy checkmark. Other names are banana decline and blackhead disease of banana. This nematode also helpful in causing Panama wilt of banana in association with fungus Fusarium oxysporium var. Cubbies. It has caused yellow disease of pepper. B. Rhodophilus atrophilus, responsible for causing spreading decline of citrus in Florida, USA 14. False root knot nematode, Nacabus sp. JRF 11, develops hypertrophy 15. Ring nematode, Criconiema sp. It has large annuli. 343p 2017. 16-pin nematode, Paratolenchus sp. This is smallest plant parasitic nematode with size of less than 0.5 mm. 17. Dagger nematode, Ziphoneema sp. Presence of characteristic sword-like stylet. It transmit fan leaf virus of grape virus. Nematode and microbes interaction. The incidence of cotton wilt, Fusarium oxysporium, was more severe in presence of root knot nematode in strawberry by the infestation of Aphelenchoides frigeri nematode and Clavibacter fusions, bacteria, cause cauliflower disease. Unit 8. Management of nematodes 1. Chemical control. A. Fumigants. CGDD. A mixture of 1,3-dichlorapropene, 1,2-dichlorapropane, pre-PG exam 2015, EDB ethylene dibromide, MBR methylbromide. Nemagon, DBCP dibromochloroPropane. B. Non-fumigants are grouped into Vi, carbamates aldecarb, temic, carbofuran, thoaden, oxymel, vidate, metham-sodium, vapum, 2, organophosphate, thionosin, nemophos, phorate, thimet, phenamifos in nemico, 2. Physical control, use of heat, hot water treatment, 54 degrees Celsius for 10 to 15 minutes. Irradiation, osmotic pressure and ultrasonic. 3. Regulatory methods, such as adoption of legislative quarantine regulations, e.g. potato tubers used as seed materials, restricted use from Nilgiri Hills, TN, to other part of country. 4. Biological control, fungus Poecilomyces lilicinus, oviparasite. Bacteria, Pasteuria penetrans, larval parasite. 5. Cultural methods. A. Crop rotation. It is most effective for moliere disease control by growing of mustard and gram. B. Trap antagonistic crop. I. Mustard. Contains allyl isothiocyanate, toxic for cyst nematode. 2. Marigold. Contains A. terthionil, reduced population of Meloidogyne spp, Paratolenchus, and Rhodolenculus. C. Flooding. It kills nematode by asphyxiation. D. Propagation through healthy planting material. 344. 2018, soil solarization in summer. It is effective by the 2nd of March. Cross deep plowing or use of plastic sheet for solarization. Unit entomopathogenic nematode, EPN. Nematode families of Steinomatidae, Heterorhabditidae have potential as biocontrol agents. Because these are entomopathogenic nematode and are facultative parasites, they are associated with symbiotic bacteria, e.g. the Xenohabdus bacteria associated with Steinonema, and the Photorhabdus bacteria is associated with nematode Heterorhabditid, e.g. Heterorhabditis indicus used against Holotrichia serrata. EPN products. In India, a commercially formulation, Ecomax, is available. It's prepared from nematode S. Kappa capsi and H. Bacteriophora. The product of Neoplectina kappa capsi with bacteria Acromobacter nematophilus is commonly named as DD136. It is also available in several labs under the trade name of Black Commando. Unit 10. Virus vector nematodes 1. Nepoviruses, nematode transmitted polyhedral particles of viruses. Tomato black ring, TBR raspberry ring spot. Longidorus elegatus grapevine fan leaf, GFLV Ziphoneema index. JIF 07, tobacco or tomato ring spot, TRSV, Longidorus sp. Ziphoneema americana 2. Netu or Tobra viruses, nematode transmitted tubular particles. Tobacco rattle virus, TRV Trichodorus similes. JIF 04, P. early browning, PEBV Trichodorus sp. Pepper ring spot virus, PRV Paratrichodorus minor, some terms mesentran of nematodes. Initoma, cuticular lining, is absent and fine finger-like projection known as microvilli present around lumen. Microvilli increases the surface area of intestine and are both secretory and absorptive in function. It provides structure like spicules, gubernaculums, both are sclerotized plates. In male, these copulatory structure are used for open the vulva to transfer of sperm. 345 2018, a competitive book of agriculture. Female ovaries, monodelphic, only one ovary, gonad, is functionally didelphic, 2 ovaries are functionally in female monoprodelphic, the female gynoduct is 1 in number and projected anteriorly. Amphidelphic in female 1 gynoduct is projected anteriorly and other posteriorly. Male testes, menorchic, only 1 testis is present, D or chic, 2 testes are present. The spicules, gubernaculums and bursa are secondary sexual organ. In male nematode, most of nematode generally possess a single genital tract is known as menorchic volcanisms, refers to number of generations in a year 1. Multivoltan nematodes, many generations are completed in a year. Examples are Meloidogyne incognita, Pratolenchus, Rhodolenculus and Tylenculus sp. 2. Univoltine, only 1 generation is completed in a year. E.g. Anguina triticea and in Heterodora sp. But Heterodora aveni is univoltine.